Good morning, students. Today we are going to discuss about the second year topic, that is the plant growth regulators, in which the first question is the applications and horticulture uses of auxins. Before going to discuss about auxins, we need to understand some basic things regarding plant growth regulators. Plant growth regulators, also called as phytohormones. So, some chemicals present in the plant body which acting as hormones, so that the hormones which called as phytohormones, they involved in growth and development of the plant body, so that they call as plant growth regulators. So plant growth regulators in the plant body are very simple molecules. These molecules are hormones which are 5 types: auxins, gibberellins, cytokinins, abscisic acid, ethylene. When we come to the derivatives of different plant growth regulators, first one is auxin. Auxin derivative means chemical. The chemical composition of auxin is indole. Indole compound. The natural name of the auxin is, or chemical name of auxin is, indole acetic acid and indole butyric acid. And the chemical composition of gibberellin is terpenes. Cytokinins, adenine, N6-4-furyl, amino, purine, and kinetin. Abscisic acid derivative is carotenoid. Ethylene is gaseous form in the plant body. Now come to the hormones. So plant growth regulators again actually classified into 2 types: plant growth promoters and plant growth inhibitors. So plant growth promoter means the hormones which useful for growth of the plant body, that hormones called as plant growth promoters. Those are inhibits the growth of the plant body, they call as plant inhibitors. So examples of plant promoters: auxins, gibberellins, cytokinins. These 3 are best examples of plant growth promoters. And plant inhibitors, best example, abscisic acid and ethylene. Now come to the first one. First plant growth promoter is auxin. So before going to discuss about the functions of auxins, some historical points need to understand. So auxins first observed by Charles Darwin and Francis Darwin in coleoptile of canary grass by using unilateral illumination. When they were used unilateral illumination over the coleoptile tip of the canary grass, the tip, they found actually some bendings into the tip of the coleoptile. And finally they said some of the factors influences present at the tip of coleoptile, so that they stimulated unilateral illumination, so the bendings occur in the coleoptile. Later the auxins was isolated by F.W. Wendt scientist from oat seedling, coleoptile of oat seedling plants. Now the auxins precursor of auxin, tryptophan and zinc ions. These 2 very important to release auxins in the plant body. If tryptophan is a type of amino acid, It helpful for the releasing of auxin, and zinc also essential for secretion releasing auxin. So hence these two called as precursor. Now auxins actually helpful for the elongation of both root and also shoot. Auxins actually of two types. One is natural auxins, second one is synthetic auxins. Natural auxin means naturally they secreted in the plant body. Examples of natural auxins: IAA and IBA. IAA means indole acetic acid, IBA means indole butyric acid. Synthetic auxins means artificially synthesized auxins in industrial level. Examples of synthetic auxins: 2,4-D means 2,4-dichlorophenoxy acetic acid, 2,4,5-T 2,4,5-trichlorophenoxy acetic acid, and NAA naphthalene acetic acid. So these are the three which best examples for synthetic auxins are artificial auxins synthesized in industrial level, and they also used as chemical fertilizers. Now come to the functions of auxins. Auxins mainly useful for the major function of auxin in plant body: elongation of cells or enlargement of cells. It means the cell size increased by influence of auxins. So where auxins present, the size of cell can be increases. And in meristematic tissue, auxins continuously secreted so that the cells of meristem continuously undergo cell division. So these are the two functions carried out by auxins. One is cell elongation, cell enlargement, and also cell division process in meristematic tissue. The second function of auxin is tropic movement. Tropic movement means movement of root system, movement of shoot system. Based on that, the movements are two types majorly: phototropism and geotropism. Phototropism means stem grow towards sunlight, that called as phototropism. So it is a type of movement towards light. And geotropism means it is occur in root system. Phototropism occur in stem, geotropism occur in root. So the growth of root is inside of the ground. That movement called as geotropism. So however, phototropism and geotropism influenced stimuli by auxins. So this is also one of the major function carried out by auxins. Now come to the third function of auxin. It can be control the xylem differentiation. So xylem again of different types, and we know very well about the function of xylem. Xylem transport water and mineral salts from the soil to the entire plant body through the root system. The other function of the auxin: apical dominance. Apical dominance means when we observe the root tip of the shoot and lateral buds. These lateral buds do not grow because of presence of apical bud. It means apical bud not influence or not help to grow the lateral buds. Whenever apical bud is present, the lateral buds do not grow. So this type of condition called as apical dominance. If you cut the apical bud, the growth takes place in lateral buds. Okay, so these are the different types of functions, and the apical dominance mainly applied in tea plantation and also hedge making. In some border plants, by hedge making method, we use auxins. Now, the another function, major function of the auxin is root initiation. So auxins promote root initiation in stem cuttings. So what type of auxins used for the promotion or promoting root initiation in the stem cuttings? Naphthalene acetic acid and indole butyric acid. As we said just now, naphthalene acetic acid is artificial auxin, IBA indole butyric acid is natural auxin. Auxins also promote flowering in pineapple by using NAA and 2,4-D. 2,4-D means 2,4-dichlorophenoxyacetic acid. Other function of the auxin is inhibition of abscission. Abscission means fall of leaf, fall of premature fruits. So when the leaves or premature fruits which drop at an early stage, that called as abscission, that prevented by or inhibited by auxins. So this is the major function of auxins. And other function of the auxin is parthenocarpy. So auxins induces parthenocarpy, mainly in tomatoes. So parthenocarpy means seedless fruits. Seeds are not found in the fruits. So that mainly induced by using auxins. Now, auxins also used during cropping as a herbicides. For example, 2,4-D. 2,4-D is a main herbicide, auxin, 2,4-dichlorophenoxyacetic acid. It kills dichotomous weedy plants during culturing of monocotyledonous plants. And this auxins also promotes healing of injuries, and it also promotes root nodule formation in leguminous plants. Thank you.